If you have seen the lessons where we dealt with the secondary growth of dicot root and dicot stem, this lesson would come as a relief from the diagrammatic representation that I gave you. You are going to find well lit and well presented diagrams of the vertical section, not vertical, pardon me, transverse section showing the secondary growth in both dicot stem and dicot root. Let's have a good representation that we have over here and a good understanding as well. What you see here is the primary growth that comes from the primary meristem. This is a dicot stem during its primary growth stages. You find phloem, you find xylem, that is the vascular bundles. There is arrangement of sclerenchyma, there is outermost epidermis, pith. We have done the structures. This is simply that representation. Now what is going to happen is that whatever lies between, the cambium that lies between xylem and phloem is going to become active and this pith is going to get crushed. So what we find innermost is the primary xylem that was formed from the primary growth. This primary xylem is being pushed inside by the newly formed xylem that is known as secondary xylem. The secondary xylem is in close contact towards the periphery with what? That is the vascular cambium. Okay. Now this line, this green line is representing the vascular cambium. We have these small projections as the primary xylem. This olive green part is the secondary xylem. This line that separates purple and green is the vascular cambium. So if the vascular cambium is giving rise to secondary xylem towards inside, what it is giving rise to towards periphery, that is secondary phloem. Okay, this is the secondary phloem. Secondary phloem in turn is pushing the previously formed primary phloem outwards. That outward pushing makes the primary phloem sit next towards secondary phloem but towards the periphery. And lastly, what do we have is, we have this region sclerenchyma that is present. In between we find cortex. This second penultimate line that you see in these concentric circles, the penultimate one represents the cork cambium. Cork cambium is giving rise to cork towards outside and inside it is forming the secondary cortex, which has not been represented over here, but you can make it out that inside over here it is the secondary cortex and outside it is the cork, and outermost one is the epidermis, which will be broken down. There could be formation of lenticels, and this epidermis till the vascular cambium, this line is known as bark. Okay, or otherwise we have the terms like phellem, phelloderm and phellogen that we have done in the previous lesson. Next is, how does secondary growth take place in a dicot root? Very simple. As you know, the arrangement inside the root is radial, alright, xylem, phloem, this such arrangement is there. Here we have the xylem, primary xylem, these ones are the phloems. Soon what will happen is a cambial ring is going to come into the scenario. That cambial ring is going to form xylem towards inside and phloem towards outside, because of which the cambial ring is going to become circular with time. What you see over here is this vascular cambium has become circular. This lighter peachish part is what? It is the secondary xylem. You see over here the cambium becomes active, it starts pushing inwards the secondary xylem and towards outwards it starts pushing the secondary phloem. So we are going to find the same arrangement, that innermost would lie the primary xylem, following it would lie the secondary xylem, and from that secondary xylem we are going to find the vascular cambium that would be present. Vascular cambium is going to give rise to secondary phloem, this light green part. This secondary phloem has crushed the primary phloem outwards. So we have secondary phloem rays which are being formed, the primary phloem has been pushed, the cortex is further pushed, and the outermost is the epidermis, the hard periderm that is being formed. So you can use these diagrams for reference for whatever we have studied, in case you didn't understand my diagrams that I made while making you explain, making you learn what we have done. So this is all that you have to keep in mind when the secondary growth in dicotyledonous root and stem is to be studied. I would suggest you to practice these diagrams so that you remember them well and you have a better understanding of the topic.